So, hi, I'm Jonathan.
I'm Jared.
And today we're going to do a perfect lesson of cells dividing.
We will now show you the process of mitosis.
Yeah. The nucleolus is in the middle of the cell, just hanging out, and the centrosomes are up here, still just hanging out, not doing anything yet. And all the DNA is just around the cell and not connected in any way. During G2 of interphase, the centrosomes right here are forming, and they will begin to create the mitotic spindle in prophase. The early mitotic spindle begins to form from the centrosomes, and the nuclear envelope begins to break down.
In the nucleus, the DNA begins to form sister chromatids as they become ready to be split apart. During prometaphase, the centrosomes grab onto the nuclear envelope as it breaks it apart and connects it to the kinetochores.
The mitotic spindle covers the entire nucleus, and the kinetochore microtubules go completely across the nuclear envelope and grab onto the chromosomes at the kinetochore.
During metaphase, the centrosomes grab onto the DNA through the
kinetochore, and the mitotic spindle is completely covering the cell now, and all of the chromosomes line up in the middle of the cell at the metaphase plate, and the chromosomes are ready to be pulled apart.
Along with the nuclear envelope dissipated in the cell, which will be rebuilt later.
In anaphase, the two sister chromatids are pulled apart into two daughter chromosomes.
Because the kinetochore microtubules are attached at the centromere, the chromosomes move centromere first. Their pace is about 1 nanometer a minute.
During telophase, the broken apart microtubules begin to form the nuclear envelope in each of the new cells, and inside the nucleus, the nucleolus begins to form.
The DNA then becomes less compact, and at the same time, the centrioles go back to their original spots. During cytokinesis, the division of the cytoplasms start to become apparent. This is when the two daughter cells with nucleus full with DNA that is not compact starts to form.
The cleavage furrow begins to pinch off and create two new daughter cells.
But that's only in animal cells. Plant cells, they have the cell plate. But yes. We will now show you the process of meiosis.
In prophase II, at the beginning of meiosis, the nuclear envelope never fully forms after cytokinesis.
And the DNA is not too compact, so these centrosomes start to make the spindle fibers as the DNA gets more compact in the nucleus that is broken.
Chromosomes are connected to the microtubules, and they are lined up across the metaphase plate.
During anaphase, the homologous chromosomes separate and move towards the centrosomes at different opposite poles. During telophase II, each pole has a new haploid chromosome set.
The microtubules break down, and the nuclear envelope begins to form in each of the new cells.
And a cleavage furrow gets built between each of the new cells.
In cytokinesis 2, the end result is now 4 daughter cells from the original 1 cell.
With variation of DNA, the DNA is now loosely packed in a nucleus, and the cleavage furrow finally breaks apart, and the cells become their own organisms, their own cells, I guess.